T helper cell activation, because we need the T helper cells for both the T killer cells and the B lymphocytes. So that's our first step for both activities. There is a page 72 of the different cytokines. I'm just going to talk about them specifically interleukin 1 and interleukin 2 in the process, rather than talking about them as separate components. So I'm actually moving ahead to page 76, and that has the steps of activating a T helper cell. Actually, yeah, T helper cell. But it's not page 7. Well, actually, you know what? That's just T killer cell. So the T helper cell is
74? No.
I had it last night. No, not 674. Let's say on page 76. No. Yeah. The T helper cell activation is shown in the picture, and then it finishes up with the steps in the sequences below, and I have the steps up here as well. Here we go. Okay. So these are the steps of activating the T helper cell, and then I have another slide for the steps of activating the T killer cell. All right. So we're going to go through the process of using the antigen presenting cell, because that's what we use to activate the T helper cell. All right. And then the T killer cell will be associated with that. So I'm going to redraw the macrophage. And we'll do the viral infection sequence again. So here's our macrophage, which is representing our APC. An APC is antigen presenting cell. Okay? So we'll draw it smaller. So we have the viral protein, or just plain virus, being endocytosed by the cell. And as we just talked about, remember it's going to interact, it's going to come into the cell, and it's going to fuse with the lysosome. And that lysosome is going to break down the viral protein into smaller components. And now those smaller components can fuse with a vesicle that has been pinched off of the endoplasmic reticulum and contains the MHC class II molecule. Okay? Fortunately, we just talked about this not too long ago. So now this will pinch off and insert its membrane into the cell, and we will have our antigen on the outside of the cell, and the MHC class II molecule. Okay. Everybody follow that through? Remember, MHC, antigen presenting cells also have MHC class I. We'll come back to that in a moment. So now this antigen is available for a specific interaction. Why do we call it specific? Because the receptor that's going to bind to it recognizes it by shape. It will only interact with other antigens that have the same complementary shape as a receptor. All right? So we're going to draw a T helper cell. And T helper cells have cell markers on their surface. These are not antibodies. They will be antibodies when we talk about the B cell, but in the T cell, helper cell and the T killer cell, these are not antibodies, but we call them T cell receptors, or TCR. And they are all identical to each other, all right? And they are unique. So another T helper cell will have another unique set of receptors. And these are randomly determined when the cell replicates, and there's a variation in the genetic expression. So we get random expressions of the genes for these T cell receptors, but all the T cell receptors on the surface of a particular T helper cell are going to recognize the same antigen. Okay? So by random chance, because there's so many of, or all of these, this T cell receptor recognizes the antigen presented by the antigen MHC class II molecule. T helper cells only interact with MHC2 molecules. So these T cell receptors will only interact with MHC class II molecules. When we get to the T killer cells, they only react with MHC class I. Okay? So an T killer cell will never interact with an MHC class II. So this molecule is stabilized by several different intracellular proteins. The only one of which you have to know is for exams anyway, is the CD4. Now if we go back a few slides, there's another couple of different ones here. So the B7 and the CD28, you may see that on the quiz. Okay, but for the exam you don't need to know those. But these are the macrophage has the B7 and the helper T cell has the CD28, and those will connect. So those are additional stabilizing co-stimulation molecules. But the CD4 is unique to the T helper cell as well. And remember I said it was the CD4 molecule that the HIV virus takes advantage of and uses as an entrance point. It can interact with that protein. All right, so now we have this interaction between the macrophage, or the antigen-presenting cell, and we're at step 3. So the macrophage releases intracellular, what we call co-stimulation. It sends, in this case, it's just stimulating the T helper cell. Let's see if I can find a different color here. Purple lead, but it's not a purple molecule. Okay. Orange. So now, because of this interaction between the CD4 and the B7 and the CD28, the macrophage releases these molecules that are internalized by the T helper cell, and these are called interleukin-1. Interleukin means between white cells. So abbreviated IL-1, which stands for actually I'm going to write that a little higher because I need this space. Now there's some variation in the cells that make it, but I'm simplifying it here. So we're going to assign interleukin-1 to the macrophage, all right? And that is what activates the T helper cell. It makes the T helper cell able to help, okay? Now, we're looking at T killer cells, so we're going to bring a T killer cell in, okay? And just for simplification here, we're going to have the T killer cell nearby. We're going to have it recognize an MHC class I on the macrophage. How would we get the same antigen that we have on our MHC class 1, MHC class 2, how would we get that same viral antigen, that same piece of the virus? It's on the MHC class 2 onto an MHC class 1. What would have to happen? MHC class 1 utilizes endogenous peptides, remember? To have an endogenous viral antigen, that virus is going to have to take over the macrophage's nucleus and get the macrophage to replicate the viral proteins. So our macrophage has now been infected by the same virus, and that antigen got into the ER and onto the MHC class 1 molecules. So let me just turn the slides back to that image to remind you. So this also happened in that same cell, okay? Just so I can have all this in the same picture. Otherwise, the T helper cell would leave and would clone and would come in contact with the same antigen on another cell and interact with the T killer cell. But here we're just having it do it on the same cell. All right? So MHC class 1. So here comes a non-reactive T killer cell. It also has T cell receptors. And these T cell receptors, just by chance, are going to be able to bind to the same antigen as the T helper cell receptors. Okay? And that has to occur for the T helper cell to be able to activate the T killer cell. So it recognizes the same antigen, and it's stabilized, but now it's stabilized with a different molecule. It's stabilized with a CD8 molecule. So let's change to the T killer cell activation, then we'll come back to this slide in between. All right, so our APC antigen is presenting that MHC-T molecule, which bound to the T helper cell. That's what we did there on the first thing on the right. Now, the MHC1 molecule is presented to the T killer cell, okay? So the T helper cell now is activated, and it's releasing its own molecules, which go to the T killer cell. And this is a different interleukin-1. It's still between white blood cells. So these molecules are interleukin-2. Finally, in sequence, numerical sequence. And interleukin-2 is going to activate the T killer cell. Okay? Now the T killer cells can multiply and clone themselves. That's that last step. The T killer cell undergoes mitotic division. Okay? So now it releases, we've got thousands of these T killer cells that recognize this viral antigen. So we're going to continue with the story. We're going to put the T killer cell up here. It's activated. So this one is now, let me do it in black, in blue. It's multiplied itself. So these are kind of roaming all over the area. It has those same T cell receptors. And now it comes in contact with another cell that was infected by the virus. Okay? This is a body cell. And so it's going to have the viral antigen antigen. And it may have different pieces of that virus on here, but if it has the same piece as what was activated that this cell recognizes, then again, by chance, the T killer cell, T cell receptor, recognizes it. Would that be an MHC class 1 or an MHC class 2? That's presenting that antigen to the T helper cell. T helper cells bind to MHC class 2. T killer cells bind to MHC class 1. Okay? Now, do you remember the two chemicals made by the T cell related cell, our NK cell? What were those two sets of chemicals? It poked holes or perforated the cell, so we call it perforate. And once it got those cells perforated, it sent in enzymes that triggered the cell to kill itself and break itself down. So our T helper cell is going to be sending perforin or perforin and granzymes. And it's going to perforate the cell membrane and send in the granzymes to trigger the cell to be killed. So that response is the same as the NK response, all right? But remember, the NK receptor is recognizing the FC, which is nonspecific, and this T cell receptor is recognizing the antigen. If it's a different antigen, it won't respond to it. That's where our specificity comes in. Okay? So the diagram that I have up here was the extra credit for the last couple of years. It essentially has what I drew up on the board. Okay? So we'll review that. So the antigen presenting cells are a macrophage. All right? What would number 1 be? So the antigen presenting cell activates this cell activates this cell, and this cell kills that cell. All right, so in sequence, here's our antigen presenting cell, and it's activating the T helper, right? So number 6 would be the T helper cell. Okay, so the T helper cells, TCRs, T cell receptors, are interacting with the antigen presented to it by the APC, and T helper cells interact with MHC1 or MHC2? 2. Okay. Well, I have 1 and 2 to get between. So 1 is MHC2, which has the antigen that the APC broke down with its lysosomal enzymes. That's recognized by the T cell receptor of the T helper cell, which is stabilized by CD4 or CD8. What do we have with T helper cells? CD4. Okay? And then the macrophage releases interleukin-1, which activates our T helper cell. Okay? Now we have the T killer cell. It interacts with the same antigen, but now that antigen is being presented by an MHC class 1, stabilized by a CD8 molecule. And now the T helper cell releases interleukin-2, which activates the T killer cell. The T killer cell doesn't have to stay bound to the macrophage. It finds that same antigen on a cell infected with the virus, same TCR, same CD8, but now the T killer cell kills that virus-infected cell through the release of perforin and granzymes. Okay? It's a simplified version, but that's the level that I want you to follow through. Okay? And this happens thousands of times because although it's random, we have the T helper cells can multiply, the T killer cells can multiply and clone themselves, so we have lots and lots and lots of those cells available. Okay? Any questions on that? All right, let's take a break. guys can study for the quiz. So I'll leave this up. You can take breaks and study for the quiz. Just let me know, you have up to an hour, okay? Or longer if you wish. I think yesterday, they got tired of studying by 40 minutes. So whenever you guys are ready, come to a consensus. You can bribe each other with candy to stay longer or shorter. Just let me know when you're ready for the quiz. Okay?
What's on the quiz?
Pardon?
What's on the quiz?
The quiz covers